This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 35, recorded on June 5th, 2012. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Howdy. How are you doing?
I'm well. How are you?
Okay, Vincent? Good. We're going to see each other pretty soon, I guess.
We're going to see each other in San Francisco.
That's a nice city to meet. And it's also the general meeting of the American Society for Microbiology, our sponsor.
Yeah.
So, and I understand you've risen in the world. You're now a big shot.
No.
You're going to be, or will be the chair of the communications committee. You think you know anything about communications?
I know how to speak. Is that communicating?
That is. Okay.
Well, no, more importantly, you know how to communicate. You not only know how to speak, but you know how to communicate.
I don't like to be chairs, but I'm going to try it. We'll see. That just means I have to go to GM every year.
Oh, you'll be
so
great.
if you're the next GM, I'll see you again.
All right. Well, that's a reason for wanting to go.
So do you just go when it's on the West Coast, Elio, now?
I go when I sort of have a better than usual reason. You know, when you're retired, you first of all, you count your pennies because I tend to have to pay for them myself.
Yeah,
I don't have a grant or anything like that. But also, sometimes I get paid because I do something, and that makes it easy. If not, I just sort of, I give myself a meeting a year. Okay, so out of my own pocket.
But you don't have anything against traveling, right? In general,
no, not yet. Not yet.
Yeah.
I don't travel as much as I used to, but I'm still okay.
That voice you heard not too long ago is also joining us today from the Medical University of South Carolina, Michael Schmidt.
Well, I'm actually in Tennessee today. I'm at.
You sound pretty good for Tennessee.
Well, I'm at the Oak Ridge National Laboratory, so they have a much better connection than the Standard Hotel. So the friendly folks at Oak Ridge National Labs are letting me pirate their internet connection, their public internet connection, so that I can have a clean shot.
And you brought your headset?
I brought my headset.
It sounds really good.
That's nice.
Very good. Well, thank you. This is a government lab, right?
This is a government lab. It's one of the DOE national laboratories.
Thank you, government of the U.S., for allowing us to communicate microbiology today. What are you doing there?
I'm participating in a meeting for one of my other tasks that I do for the Department of Homeland Security. I manage a number of education programs for them, and we're having our annual conference where the graduate fellows get together and the postdocs get together and they look at opportunities at the national laboratories. So it's a meeting that's sponsored by DHS, and we're holding it at in Oak Ridge. So it's a great thing. They talk about all sorts of really neat science that's well above my head, invoking things like quantum mechanics and other things, and so it's a really neat meeting for them.
Nice. Well, good. I'm glad you could take an hour to join us.
Yeah, that's nice.
We have two interesting stories today, and the first one was suggested by you, Michael, not too long ago. So we're picking up on your suggestion.
Well, that's good, and I thank you for that. It's this is a tragic story that's been recently in the news media. And it's specifically about this young woman, 24 years old, who has been battling a rare variant of a flesh-eating bacterium she contracted after cutting herself when she fell from a homemade zip line. And zip lining is sort of like parachuting without the parachute. You sort of, attach yourself to a line and you slide down this line. And unfortunately, the zip line broke and she fell and she got a big gash in her leg, and she was inoculated with a bacterium that's indigenous to fresh water and slightly brackish waters. And this particular bacterium is Aeromonas hydrophilia. And unfortunately, this particular microorganism is resistant to many antibiotics. So when it penetrates and gets into your tissue, it develops a form of necrotizing fasciitis, where, just as its name implies, necrotizing, where it looks like it's rotten, and the fascia, where it's effectively rotting the flesh that's associated with this. And this poor young woman has this disease has now claimed her left leg and will likely take her fingers as well. She was in a coma for quite some time. And so I've been stopped in the hall at work, and people are very much concerned about this, because a lot of us in the Southeast go swimming, we go ziplining, we do all these things that this active 24-year-old woman was doing, and we're, of course, very much concerned about the microbial world attacking us. And I think what this story teaches us is many important lessons, and we're not going to get into the medicine, because the medicine of this disease process is pretty straightforward in how you treat it. It's really old-time surgery, where you just literally take away the dead tissue until you see good, clean tissue, and you hope you get it all, so that
what's the word for that?
Debridement or debulking, or
Okay. That's good enough.
Yeah, it's good. I just wanted you to say debride, not debride.
No, debriding. My good friend is a surgeon, so that's been beaten out of me.
I was once told, just don't think of brides, and you'll get it right. Debride. So why do you have to cut it away? Is it?
Well,
it's just dead flesh.
This is what I found absolutely fascinating about this is because the microbe's still in there. The microbe is still in there, and you got to get really to clean margins. And it's the hallmark of all cancer therapy is they look for clean margins of when there's healthy tissue. And we've all seen enough medical TV shows to know when they quickly go and get a frozen section and take it to pathology to see if there's any invaders in there of cancer cells and whatnot. But I think what this story is going to teach us is all bacterial disease, but especially all invasive forms of bacterial disease, requires the expression and host response to the bacterium's expression of a variety of virulence factors. And when this particular microbe, Aeromonas, was introduced deep into her tissue from this big gash, she had, I think, if I'm remembering correctly, 22 staples in her leg to seal the gash. This wasn't a little cut that you had. This was a deep wound that required staples. And anytime I hear staples, I sort of get a little squeamish of thinking about how big of an opening that is, and just thinking about the concentration of microbes that are in normal fresh water. That could be auto-inoculated into that wound.
But they didn't have any. They hadn't have any clue when they stapled her that this was going to happen.
No, there was no signs. And the standard of care is you will treat her prophylactically with a cocktail of antibiotics, and typically they'll flush the wound with an antimicrobial cocktail of the drugs of choice that are moving around, that drugs of choice that are effective against the bacteria that are moving around the community at the time. The antibiogram is your best friend when you have a big gaping wound.
A couple of questions that arise immediately. You're probably going to go into it. So Aeromonas is a bug that's found in brackish waters, and it causes big-time ulcers in fish, doesn't it?
Yes.
But it's
absolutely
not usually associated with much of human disease, is it?
No, this is a rare event, and there's I learned a lot about necrotizing fasciitis because what I'm gonna focus my talk on are virulence factors towards the bulk of this, looking at the streptococcal virulence factors because most is known about it, and there's some really interesting parallels here. But there are three types of necrotizing fasciitis. The first type is a polymicrobial infection, where we see at least one anaerobe and one facultative anaerobe, either from the Enterobacteriaceae family, or it could also be a non-group A streptococci. Typically, the second type of necrotizing fasciitis, or the abbreviation fans will call it NF, is the most frequent variant of this disease, and that's caused by group A streptococci, the good old gas bug, which everyone in America has probably heard of because this is the classic microbe that causes strep throat. This is the beta hemolytic form of group A streptococci, which is responsible for approximately 10 million infections per year, where between 9 and 11,500, according to the CDC, result in invasive forms of this disease, which either manifest themselves as streptococcal toxic shock syndrome, which is very similar to the disease process we see with staphylococcal toxic shock syndrome that was associated with the tampon outbreak back in the 80s when they had those highly absorbent tampons. And the third form is, I think this is the parallelism of TWiM. The third type of necrotizing fasciitis is from marine vibrios. And of course, the most famous marine vibrio that causes the NF is Vibrio vulnificus, where it literally looks like you've taken your arm and you've dipped it into a vat of boiling oil, because your arm literally looks like it's gone through a volcano and it's vulcanized. And that form of disease, the first one.
I never heard of vulcanized used in connection with human disease, but it's very apt. It's a good choice of words.
It's a really good description because it's the vulcanization process or the fulminant necrosis takes place so quickly. It looks like you literally got hit with a volcano on that extremity. And there's some really interesting parallelisms. I posted in Vincent's show notes the one paper I found on Aeromonas that's been reported in the clinical literature. Obviously, this is too new of a story to have made it into the clinical literature. And this particular paper was published, I'm trying to look for it, it was published in a British journal, specifically caused by an inconspicuous infection of Aeromonas hydrophilia in an immunocompromised host. And this is a man who's over 60 years old. And when you look at the graphic pictures in this article, you will quickly appreciate why surgery needs to be done, and they move in to get rid of as much of the diseased tissue as possible, and they go to clear it up. So Aeromonas just so happened to be in the pond that this poor woman fell into, and she's been auto-inoculated with this particular bug. And I think what the news media is making out, and they're not quite getting it right in the sense that they're just talking about NF and they're not really making the distinction about Aeromonas here. They then go into the statistics of the rarity of necrotizing fasciitis.
So how rare is it? I was going to ask you.
It is. This is a rare disease. It doesn't occur that frequently. As I said, it's effectively one in. It's effectively between 500 to 1,000 cases per year in
Worldwide
the U.S.
or in the States or where?
In the U.S. In the U.S. And in fact, in the Nature review that I'm going to be specifically talking about, in the case of streptococcal NF, the incidence rate per 100,000 people is 0.25 or 25 people per 100,000. But the unfortunate thing, and this seems to hold true for all three variants of NF, whether it be a polymicrobial infection like we're probably seeing with this young woman, or in the case of the more classic NF that's, we see the most of in the United States each year and worldwide for that matter, the rate seems to hold true at about between 0.2 and almost 0.1, with the exception of New Caledonia, for which some reason New Caledonia has an incident rate of 17.3 per 100,000 per year, and that
Still pretty low.
It's still pretty low. It's very low, and this then begins to teach us about the biology of the streptococcal form of the disease, and this is this review in Nature is by two of your colleagues at four of your colleagues at the University of San Diego.
California,
University of California.
There is a University of San Diego, which is different.
It's University of California, San Diego, La Jolla, in the Department of Pediatrics. And what I'm going to be talking about is this molecular insights into invasive group A streptococcal disease by Jason Cole, Timothy Barnett, Victor Nizet, and Mark.
Nizet. He calls himself Nizet.
Oh, Nizet. The French, which I haven't.
Well, he's of French origin.
And Mark Walker. It's probably like Stephen Colbert. Stephen Colbert was a native of Charleston, South Carolina, and when he was going to school in Charleston, South Carolina, he was Stephen Colbert. And the story goes from one of my colleagues' wives, who was his high school English teacher. Stephen Colbert was lamenting about how he was ordinary, and Mrs. Wise told him that he could just use the French pronunciation of his name to make himself extraordinary. So henceforth, he called himself Stephen Colbert instead of Stephen Colbert. His father was the vice president and provost of our institution, and he and two of his brothers tragically died in a plane crash September 11th. Not the famous September 11th, but nevertheless, it was a September 11th, and it was plane crash in Charlotte, North Carolina, and it's an unfortunate thing. But Stephen Colbert calls Charleston home, or did call Charleston home, and he did the.
You're giving your town a nice plug there, aren't you? Should we come visit you?
You can come visit. We have Chamber of Commerce weather. We did. It's now moving into summer, and we've already had our second hurricane, our second tropical depression, Beryl.
Listen,
why is it called flesh-eating disease?
Well, let me ask you. Let's go back a little bit.
Okay.
Streptococcal disease has sort of interesting histories. In the 19th century, one of the major diseases to affect humans was scarlet fever, caused by Streptococcus. Now, you never hear of scarlet fever. No, I'm not sure I know why. Do you know why, Michael?
I think it has to do with phage. And I think the phage and the toxin have been depleted from the population, which also may offer an explanation of the New Caledonia. And in fact, that's what this particular paper goes into, is the presence of this prophage. And the prophage they refer to as M1 T1, and this bacteriophage encodes a whole slew of virulence factors in this particular microorganism. And when you have M1 T1, because it has this particular prophage in it, the net consequence is this transitions this streptococcal disease from being a normally non-invasive form, which is, strep throat. Strep throat is bad enough because you can, of course, have non-suppurative sequelae like rheumatic fever.
Wait a minute, that's a big word. Say what it means. Non-suppurative.
Non-suppurative is, well, the literal definition is without pus, but the way I always interpret it is means without infection, where your immune system is actually reacting to your body's perception of seeing a new streptococcus come in.
Well, some of the antigens stay around for a long time.
Yes, that
so
too.
The bug is gone, but the antigens are still present. Therefore, the body responds to that. Isn't that right?
That is correct. And that's particularly apparent with one of the virulence factors associated with streptococci, and that happens to be the famous M protein, which was described originally by Rebecca Lansfield during the first part of the 20th century. In Lansfield grouping of streptococci, that's where we get group A, group B, group C, etc. is the bane of every medical student's existence of memorizing these things. But the M protein is really a neat protein because not only does it look similar to one of our own cardiac antigens, but what this sucker does is it mediates adhesion to host epithelial cells. And when you mediate adhesion to the host epithelial cells, this facilitates resistance to opsonic phagocytosis through the binding of fibrinogen and a complement inhibitory factor, H, and the C4b binding protein and a number of immunoglobulin Fc regions. And the net consequence of that M protein being able to adhere to the epithelial cells is it then increases survival in neutrophils and something called neutrophil entrapment nets or nets. And this net stuff is really pretty neat, these extracellular traps. And if you think about what the life of a neutrophil is about, it effectively employs two strategies to kill invading pathogens. It first wants to eat them, and the second thing is it secretes antimicrobials. And so this then will tie us into the story that we're going to talk about with the Vibrios and antimicrobial peptides and the other things. So back onto the topic is
you're going to answer my question,
I think I forgot.
I'm waiting here. Flesh-eating bacteria.
Oh, it's because of where these microbes attack. And it's effectively, literally, the fascia or your skin. And so
the covering of the muscles, isn't it? What fascia is. That's what it is. And the reason I think that the bug likes it is because that's sort of not a well-vascularized site. It's really just a sheet which covers the muscles. There's no blood vessels in there. Of course, there's some liquid in it, but it's just a covering. And underneath that covering, sort of it's pig heaven for the bugs because they got a lot of nutrient and they have almost no defenses there. The body has almost no defenses there. It has to recruit them. But it's a very sheltered environment, actually, the subfascia.
It's really the best niche for these things. And this particular phage, and for this here's where it really the streptococcal paper got really interesting for me. And this is what I also am hoping the lead investigators at the CDC who are looking at this Eromonas outbreak is they ask the question whether or not there's a phage system in the Eromonas that's very similar to the phage system that's responsible for the virulence of the streptococcal disease, because they're still eating the same food source. And microbes are really clever at exchanging information amongst themselves, especially when it's doing sort of the same thing. And so the authors from San Diego, from UC San Diego, really talked interestingly about this CoVAR-S two-component system that results in a mutation in this two-component regulatory system that then results in the strong transcriptional upregulation of multiple virulence-associated genes. And the first gene that you begin to look at that's upregulated in this system is you look at streptolysin O. And streptolysin O is this cholesterol-dependent cytolysin that basically pokes holes in our host cell membranes. And when we begin to poke holes in our membranes, things leak out and it begins to feed the microbe. And at the same time that streptolysin O is being transcribed, another virulence gene is co-transcribed, and this is NAD glycohydrolase, which then actively translocates into the host cells by streptolysin O. And so all of these things are getting together, and you begin to see and there's a whole slew of other virulence factors, including interleukin 8, another protease, and a DNase, that then the simple upregulation of the DNase, which is SDA1, allows group A strep to escape neutrophil-mediated killing and persist at the initial site of infection. So you again begin to see this ramping up of streptolysin O. You begin to see leaky membranes. And because you have this DNase and the neutrophil extracellular traps, which are effectively these nets or neutrophil extracellular traps, contain DNA, they contain histones, they contain granuloproteases and antimicrobial peptides to kill bacteria. But the bacterium has all of these remarkable virulence determinants that are effectively taking out the very thing that is supposed to protect the fascia. And this one phage then has effectively, and the one mutation in this two-component signaling system has effectively made this hyper-virulent form. And they're reporting that it's this M1 T1 variant because of this particular phage. And then they also have found another virulent variant that contains an additional prophage, and this one's named Z, that also encodes more virulence factors that then facilitates the production of a superantigen, SPA. And the digression about superantigens is typically a normal antigen induces a T cell response where about 1 in 1,000 to 1 in 10,000 T cells are activated. But superantigens typically
in a specific way, that is, leads to the formation of specific antibodies. Right?
Specific antibodies, whereas in a superantigen, it binds outside of the MHC groove. You activate 1 in 5 of the body's T cells, and then you create this massive, excessive, uncoordinated release of cytokines, which I refer to.
But also, you make a huge amount of antibodies, which are against nothing. They're
That's
not
right.
Against the bug that set this off, right? So you're wasting your antibody making capacity, making antibodies that are totally irrelevant to the situation.
And that is the hallmark of I think the overall pathogenesis of this is where you begin to have these superantigens and you see the ramping up, and why this disease process can be so aggressive and so fulminant, and why simple antibiotics, which are normally quite effective at treating normal infections, just don't work. I mean,
let's go back. Hold on. Before we talk about therapy, why don't we stay with this for a moment? Because what you said about one phage and then the other phage is probably the key for why strep is such a variable bug in terms of pathogenesis. In other words, historically, we had the case of scarlet fever. It's true, by the way, that this kind of fasciitis, this flesh-eating condition, seems to be fairly constant since we kept records of this sort for the last 100 years. So, but on the other hand, scarlet fever and strep throat itself and rheumatic fever seem to oscillate in the population. They're not a stable disease. And what you said seems to be the answer to that. Namely, it takes phages to make these bugs into the virulence guys that they are. So I think that's a nice story.
That was my take-home message for the folks working on this Eromonas outbreak. I would really love them to hunt for phage in this Eromonas, because I think you'll be able to pull out a phage or a phage-like story that we see similarly in this streptococcal story, because we haven't heard about Eromonas causing necrotizing fasciitis. I mean, the other microbiologists that I routinely interact with, we all scratched our heads when we heard it was Eromonas, because to a person, we would have all bet cash money that it was probably a streptococcal one, until we actually saw the reports coming out that it was indeed Eromonas.
So, Michael, how do we know that it is Eromonas? Just because they cultured it from the wound?
I think that is true. I've tried to see if CDC has published it in MMWR, but I have not found that. I found that one CNN story where they reported it as Eromonas, and that is what I am going by. Again, as I said,
but wait, it's not. This is not unheard of. This is in fact the third kind of fasciitis. It's the Eromonas one has been known to happen before, so it's rare. But I wouldn't be so surprised that you have a case.
Okay, so Eromonas fasciitis has been described.
Yeah, it has. Sure. So it's
okay.
not. Okay. Yeah, it's not that surprising. It's unusual, but not unheard of.
Okay.
I pulled a paper out of the literature, and what typically predisposes you to developing this disease is the classic things that predispose you to almost everything, whether or not you're immune compromised, whether or not you have chronic health problems. And for the case of streptococcal NF, there's a strong correlation with having diabetes, or if you have cancer or liver or kidney disease. The other thing that predisposes you is cuts in your skin, especially deep cuts, including surgical wounds. And then the other really interesting thing is recent chickenpox or other viral infections that cause a rash. And I found that to be interesting, but not really when you begin to consider what's going on with a rash, where you may be able to feed the microbes that are now in this new niche where you may actually have a rash. And of course, the final thing is where you suppress your immune system by using steroids. And so it's
Michael, do most of us harbor group A streptococci on our skins?
We do it one time or another in our throat, don't we, Michael?
We harbor them in our throat, but not so much on our skin. I don't know what the general incidence in the population on skin is.
Okay. So when people cut themselves, get a deep puncture or something, is it the case that they're inoculating themselves with some group-A strep from themselves, and that's what's causing the problem?
It's most likely the problem, but what this one review is arguing is you have to have the streptococcus that has this particular phage resident. You need the M1 T1 phage, and then for the especially aggressive form, you need the second prophage that is then going to be responsible for the next suite of virulence expression.
So if you happen to be unlucky in carrying that strain with these phages, then you're more likely to get this necrotizing fasciitis, I suppose. Because you could also be harboring a strain without these phages, right? And then they should be inconsequential.
Correct.
Has anyone done a survey of the population to see, who of those people carrying group A strep in their throat, how many have the phage and how many don't? Probably not, right?
I wouldn't be surprised. Do you know, Michael?
I'm trying to remember if I read it in the review, but I don't believe I'm looking to see if clinical epidemiology.
This, by the way, would be a neat exercise in in the medical school course. In most medical schools, the only laboratory left is in the microbiology course. All others have given up on labs. But most people still hold on to teaching microbiology with a lab. Do they at your university?
No,
Vincent,
it died with the hurricane.
Yeah, ours too. Although, so we don't have any labs, but I was at Mount Sinai last week, and they still do labs there.
Well, at Tufts, I spent most of my life, we used to have the students grow bugs from their own throat, and about 10-20% had beta hemolytic streptococci. So this was pretty dramatic. They had staph, they had all kinds of things. And it would be a great exercise in those medical schools that still hold on for dear life to the fact that microbiology can best be taught in connection with the lab to maybe check for the phage. It wouldn't be impossible to do in a class.
That's
it's
right.
a doable experiment.
And I think what we're learning as we started this discussion with the reference to scarlet fever, I mean, this is really important in the story from last time when we were talking about the algae with effectively having viruses being introduced into these phytoplankton and responsible for the remodeling of these phytoplankton. It's really no different than what we're seeing in the remodeling of these pathogens. So viruses, I know this is not This Week in Virology, but viruses are extremely important at the whole population dynamic of microbes and how they interact with people. And it's really, I think, an important lesson. And it's a really inexpensive way to really drive home the importance of understanding these sorts of things, to just ask how many things have phage and how you can get them to come out, and you learn the wonder of shining a UV light on a population and actually seeing what actually happens. And then you can explain why UV light actually causes a prophage to leave a cell.
This is really a wonderful review. They have such a list of virulence factors for these group A streps. They have wonderful pictures that summarize them too. So if you, I don't know, this is probably not open to most people without subscriptions, but if you do, check it out. It's really nice. I really thought it was very comprehensive. Is that about it,
That's
Michael?
about it. It's an emerging story, and stay tuned, as Elio would say, because I think as this young lady gets out of the hospital, God willing, she I think will be able to understand better why a simple accident from a zip line resulted in such a horrific outcome for her.
Although this, many other people could have the same injury and they wouldn't get the same infection.
Correct.
So that's the point. It's very rare. And maybe there is something about her particular genetic makeup together with the bacterium that caused this.
It's very possible.
That's very likely, but we're not there yet. Well, there is a connection between this paper and our next one, which I just realized, and that's Vibrio.
Right.
And I didn't think of that when I picked up this second paper, but now I see it. That type 3 fasciitis there, Michael, is Vibrio, marine Vibrio.
Yeah, I thought it was intentional on your part, especially with the antimicrobial peptides.
Right.
No, I'm not that good, Michael.
Well, I don't know that Vibrio cholera and Vibrio and Aeromonas are that, well, they're related. I just don't know whether the taxonomists would say about them, but they're certainly both waterborne bugs. And what Michael said, the story of the antimicrobial peptides does unite them with the one we just talked about. So yeah, there's a connection.
So the second paper was just published in PNAS. It's called Amino Acid Addition to Vibrio Cholera LPS Establishes a Link Between Surface Remodeling in Gram-positive and Gram-negative Bacteria. And perhaps it was my ignorance, but I found this really interesting, and I learned a lot. I hope you liked it.
Oh, sure. It's a good paper, and it's a relatively straightforward paper. Let me see what the context is for this paper. bacteria face an awful lot of antimicrobial activities in and out of the body, and they respond to that. They respond to that by devising mechanisms to make them resistant. That's obvious with antibiotics, and it's maybe not as widely known that we have a tremendous number of chemicals in our body, which we make, which are defensive chemicals. They tend to be small molecular weight peptides, and that we're full of them, and the bacteria have to do something about it. They also, this introduces two subjects. One is the subject of these antimicrobial peptides. I'll talk about this a little bit. And the second subject is how do the bugs become resistant to it? And just to anticipate what I'm going to tell you, which you know, but the listeners don't quite yet know, is that this has to do with substances on the surface of the bacteria. Like in the case of gram-negatives, a thing called lipopolysaccharides. I'll explain about that. In the case of gram-positives, other compounds, all of which get modified by, oddly, the addition of amino acids. And that this leads to resistance to antimicrobial agents. So that's the story. I'll start at the beginning now. Antimicrobial agents that I just mentioned, the small peptides, are made by everything. They're made by humans, they're made by all animals, they're made by insects, flies, whatever, and they're made by bacteria themselves. You may know them as bacteriocins. That's the name. Originally, the name was given to colicins because they came from E. coli, but many other bacteria make small peptides which affect other peptides. The ones that make it usually have an immunity system, so they don't commit suicide by making these antibacterial compounds. Anyhow, you know them in some connections, in some therapeutic connections. I think many readers will have used polymyxin, a triple antibiotic ointment for skin lesions, for skin cuts, and so forth. Well, two of those antibiotics, I'm not sure it's in that formulation, but in some others, can be polymyxin and bacitracin, both of which are made by bacteria. So bacteria make compounds which are antibacterial. We make compounds which are antibacterial. They work in a variety of ways. The main ones are to poke holes in the membrane of bacteria, just like Michael talked about in relation to some of the streptolysin, the toxin made by Streptococcus. The other is to collapse the proton motive force, that is the energized state of the membrane. And when that happens, the membrane doesn't work as a membrane anymore. The cells become totally permeable, and that can lead to death. And the other is for some of these peptides, oddly, to penetrate into the bacteria and inhibit the synthesis of DNA, RNA, proteins, okay, and other things. So there's a huge armamentarium here that bacteria have and that we have, making a tremendous number of compounds. There are probably hundreds of different AMPs, antimicrobial peptides. Okay, so that's the first thing to know, that they are they're found all over, and that they lead to the question: do they actually have more therapeutic value than what I just mentioned, the ones that are used in ointments? Well, the answer is it's all under study. And there is a paper by Vincent, the paper by Hancock. Maybe you could put that in the show notes. It's a beautiful paper that describes in great detail the immense variety of attempts that are being made to make effective antimicrobials, being that we're in an age where classical antibiotics may have lost their potency due to drug resistance. So it's a big subject, okay? So part of it is the antimicrobial peptides. By the way, they are chemically interesting. Some are cyclic, some have the amino acids in them, stuff like that. They're interesting compounds. Where you draw the line between a peptide and a protein is a matter of your own interpretation. We have one of the oldest, actually the story goes back to Mechnikov, the guy who discovered phagocytosis. And he asked the question, why is it that the eyes which are bathed with bacteria. After all, we're giving bacteria in the air. Our eyes get hit by bacteria. And of course, we have eyelids they're like window washers in a car, but that's only part of it. So sooner or later, people discovered lysozyme. I think that Fleming, actually, the discoverer of penicillin, was responsible for it. This is a small molecular protein. You can call it a peptide if you want to, which is antibacterial, and it works by dissolving the cell membrane that practically all bacteria have. So that's the side of the antimicrobial peptides, the AMPs, okay? So now let's ask, how do bacteria respond to that? Well, this paper picks up on the fact that gram-negative bacteria and gram-positive bacteria both do it, and they do it by a mechanism which is biochemically similar but deals with different targets. Gram-negative bacteria are characterized by having two sets of membranes, in the cytoplasmic membrane, I'm sure many of the listeners know this, and an outer membrane. The outer membrane is made up of two leaflets, just like a regular membrane should be, but the outer leaflet is not made up of phospholipids, which is what you think of normally as the lipids that make up bacterial membranes, but of compounds called lipopolysaccharides, LPS, also known as endotoxins, also known in part of them as O antigens. These are compounds which are not made of phospholipids, but are made of sugar lipids with phosphate groups analogous to them. In other words, glycolipids. These are the reason for this seems to be that it makes the bacteria much tougher. It's a defense mechanism of the inner membrane. The cytoplasmic membrane in all bacteria is sort of a weak link. Detergents or the hydrophobic compounds can really hit them. And the gram-positives defend themselves by making a huge layer of peptidoglycan, the cell wall material, which is a amino acid sugar compound, and that protects the cytoplasmic membrane. In the gram-negatives, the layer of peptidoglycan is quite thin, so that's not a defense mechanism. So they make an extra membrane, and the extra membrane, in order to be effective, has properties to defend itself against lipids. So the lipopolysaccharide outer leaflet of the outer membrane, forgive me, that's a mouthful, is a defense mechanism that these bugs have. Now, lipopolysaccharide is, as I say, is made up of sugar moiety, lipids attached to it, and attached to it in addition is a big long chain of sugars called the O antigen. I bring that up because we're talking about Vibrio here, but it's very similar to Salmonella and E. coli, where these antigens, these O antigens, are used for typing and for determining what strain you have. So when you talk about an E. coli O157H7, you're saying it has the 157 variety of the O antigen. Now let me make a digression. Do you happen to know what O and H stand for in 157H7? Either of you?
The H is for flagella.
Great, you passed the exam. and the O is, as I just said. So let me tell you where this comes from. You want to know, but now, do you know what this means? Why the letters O and the letter H? Where does that come from? Aha. Gotcha.
That I don't know.
Okay. It comes from the German. The word O means ohne, without, hauch, H-A-U-C-H, whereas the H means hauch. And hauch is a term that's difficult. It refers to the following. Some Salmonella, when you put them on very soft agar, this agar that doesn't have quite as much, it has a bit more water than usual, they will swarm. This is a flagella motility, and this is an indication that they have flagella. And the H antigen, as Michael properly said, is made up of the flagella. It's the flagellin protein is the H antigen. So they make a hauch. Now, what's a hauch? A hauch is difficult to explain what it is because I can't find a real proper English word for it. I speak some German, but it's a strange term. A hauch is what happens when you breathe on a glass surface. You have a film against it. That's a hauch. Okay, what would you call it? A film?
You would call
An
it the
emanation?
morning shower. You call it your morning shower when the glass is all fogged up.
Fog up.
Well,
Yeah.
that's a hauch. So with hauch is H, and without hauch, ohne hauch, is O. Sorry for the digression.
No, that's a good one.
This was
I like
used
that.
in the 1930s, 20s, and 30s to classify various kinds of salmonella. It also works for coli. It works for other things as well. So the combination, you have hundreds of combinations of O and H, and the permutations are huge in number. And so,
So the own is OHNE.
right,
Is that right?
OHNE, Ohne,
And hauch is HOUCK.
CH,
HOUCH.
hauch,
Okay, thank you.
hauch.
I have to put that in. Without actually, they define it without film.
Without
So they say hauch is
Okay.
film.
All
Good.
right.
So
Anyhow.
does that mean it goes back to gliding motility, maybe?
No. This is very different. Gliding
Okay.
motility is a very mysterious kind of motility that people haven't quite worked out. It is not flagellar motility. This is swarming, which is what Proteus does in some Pseudomonas.
Yeah.
It's regular agar. But most enteric bacteria like E. coli and Salmonella do it if the agar is a little bit less heavy, less dense. And so this is flagellar motility. And we ought to talk about that someday because it's a fascinating subject, and it's one that I've been personally concerned with at one point. Anyhow, let's go back to this now. Now we've sorted out that there are components of gram-negative bacteria called the LPS, which have this O antigen. It was a long digression. I'm sorry, but let's go back to it. So it turns out that if you look at Vibrio cholera, the agent of cholera, you find that there is a strain which was the one that did the great pandemics over the years, and it's called the classic strain, and it's called O1. That strain has been substituted by another strain called L-Tor. That's capital E-L, new word, capital T-O-R. L-Tor, by the way, I looked it up, is a town in the tip of the Sinai Peninsula. And the reason it's called that is because pilgrims who came back from the Hajj, the pilgrimage to Mecca, came down with a form of cholera, and people realized that this was a different strain. It's different from the classic strain in many ways. But one of the ways is that, and this has been used diagnostically, the classical strain is sensitive to the antibiotic polymyxin. Again, one of the AMPs, the antimicrobial peptides made by another bacterium. And the L-Tor strain is resistant. Okay, so the question has come up for a long time. Why is it? And it turns out that this is not only true for polymyxin, it's true for other antimicrobial peptides of the cationic variety. Okay, so what's the story? If you look at the two strains, the classic strain and the L-Tor strain, they differ in that their LPS, in the case of L-Tor, contains extra glycines. Okay, strange. An addition of glycine should make this much difference, but it does. And it turns out, this is the paper that describes it. Turns out that not only is there one glycine added, but in some cases it's two glycines. There are three proteins involved, called ALMs. And this is a story which is similar, analogous to a story which happens in the gram positives. Now, in the gram positives, again, there is the addition not of glycine, but of alanine to target compounds. The target compounds here, I'm sorry, but we're really overloading this show with a lot of terminology, but there are in gram positives compounds besides the peptidoglycan that I referred to earlier, compounds called teichoic acids. And teichoic acids are involved in the maintenance of the structural maintenance of the bacteria. They're also involved in sticking of the bacteria, adhering of the bacteria to surfaces such as cells.
They're part of the that big peptidoglycan
They
layer.
are
Yes.
that's right. They're embedded in the peptidoglycan layer. Anyhow, so teichoic acids, when alanylated. That is when an alanine is added to them, become resistant to a whole variety of antimicrobial peptides. So the story is analogous here. In both cases, you are adding an amino acid to a structure which is important in pathogenesis. And in both cases, that modification, relatively simple modification, gives a change in the ability to resist these antimicrobial peptides. Now, the reason is the following. The surface of bacteria is normally quite negative, and that is a good place for cationic, positively charged antimicrobial peptides to go and sit. But if you add glycine, you make the surface of the bacteria more positive. And the electrostatic business of negative and positive liking each other goes away. So the apparent reason is very simple. The reason the bacteria become resistant to these antimicrobial peptides is because they change their electrostatic status on the surface.
That's amazingly
Isn't
simple,
it amazingly
right?
simple?
I'm always believing that it can't be that simple, right? Just a charge difference.
It sounds
amazing.
insane.
It's amazing.
Amazing. Yeah.
Yeah. Now, this difference between the cholera biotypes, this happened in nature, right? We didn't do this. So it must be that they encounter these peptides in nature and therefore have acquired the ability to be resistant to it.
Or at least react.
Right.
Yeah, because we don't use polymyxin to treat cholera, right?
Not likely, but there is plenty. When Vibrio cholera gets into the gut, it finds a whole slew of antimicrobial peptides because all our tissues make them, and all our secretory tissues make them. So the epithelial cells are full of them, and so they encounter these guys. And since not all these peptides are cationic, but many are. The majority are. So they figured out, gee, send me something that's positively charged, I'll go positive myself.
Right. Now, I think that's an interesting question. What was the selection that led to this displacement, of the classical with this Altor?
Not that in the paper, actually. And
Could it be something as simple as the phage receptor? Because I know that phage control Vibrio cholera outbreaks, and it's a phage dynamic that's going back and forth. So could it be something as simple as changing the receptor, and it's facilitated by the emergence of this resistance, and it's sort of back and forth between the phage that goes from the epidemic form of Vibrio cholera to the normal steady state that's found in the water systems?
I like it. I think it's a good point. I don't think anybody knows. In fact, I think it's surprising that not more is known about it, but I suspect that people are going to work on it and they're going to find out. And we may have another TWiM on that one of these days.
Do you want to comment about the fact that Vibrio cholera has 2 chromosomes, and one of the chromosomes looks very similar to gram-positives?
Yeah, I didn't look that up, but I have to do it from memory.
I began to scratch my head and began to wonder about that, but then I got lost in the methods. I very much enjoyed the Maldi-Toff methodology, so I sort of got distracted by the new method of looking at these sugars, and got
very fancy stuff.
Got sidetracked by it. But
this is just to say it. Let's see, what is it? The methods that they use is not just mass spec, but a thing called UVPD mass spec, which is ultraviolet
photodissociation.
Photodissociation, right. I know nothing about that, so I couldn't tell you. But it takes fancy chemistry to figure out what's going on, or at least to figure it out in a rapid way. So there are techniques to study these things which are really extremely effective.
What I liked here was they figured out using this UVPD that there were extra glycines on the lipid A moiety, right? And then they just looked in the genome and said what genes could put something on lipid A. And they found 3, and they mutated them, and they said these are the genes responsible for sticking
well, they have nice data for that.
It's beautiful. Yeah. And then, of course, when you mutate those genes, these are lipid A acyltransferases, then they become sensitive to...
What they don't talk about is whether these are really... They look like they're constitutively made. And
yes, that's the question,
I don't think that's dealt with in this paper, but I would...
So you would predict they might be induced when you put
the
compound.
Yeah. I mean...
Yeah. There was the one in your blog that you had a post about C. diff, which is has a similar story, modification of...
This teichoic acid.
Teichoic acid. And there, the enzymes are induced, I believe.
That's right.
If I remember.
Yeah, I think that's what it said on the blog.
Yeah, okay.
Yeah. So we'll put a link to that. It's very interesting. So does the fact that the gram-positives and negatives have a similar approach modify their outer, whatever's on the outside of them? That's consistent with them coming from a common ancestor, right?
Well, yeah, but it seems to me that these are not what's involved are different enzymes. They're not conserved.
Okay.
So yeah, I wouldn't say that. I mean, I would say the opposite in a way. That when these guys encountered antibiotics, they both developed their own strategy, and it turned out to be mechanistically similar, but in detail it's different in terms of what amino acid they put on, what proteins are involved. So I'm not sure this is
okay. And so because they both have negative charged outer layers, so that mechanistically you just want to neutralize that, right? Okay, I got it. And the other thing, from my naive viewpoint, you were saying earlier, you were describing the structure of a gram-positive versus a gram-negative. I mean, to me, it looks like they could be from a common ancestor because the gram-negatives have a little bit of peptidoglycan between the two membranes, right? So you could see that as a I don't know which came first.
Oh, boy. This is a heavy-duty subject.
Is it really?
Yes, absolutely. Look, when you look at the tree of life and you see a nice branches, you see 3 branches, 3 domains of life, you look at each one of the branches, the sticks coming out, okay? Branches coming out. Just what happens at the origin of those branches is all speculation. And it's not known who came first, gram-positives or gram-negatives, and how this happened. This is a mess right now. I think phylogeny is not as developed as you may think.
So the addition of glycine or alanine, does it happen early in the synthesis of these components and then they're shipped out, or does it happen
I think it happens afterwards. I think it's something, I think it's added once they're made. Be hard to do. I mean, well, these guys are made on a membrane. And I don't know. Actually, it's a good question.
So they have a model here in this paper that says that the alanines are added on the inner membrane. Then these components get shipped out to the outer membrane, right through the periplasm.
Right. Well, there's a periplasm in gram-negatives. Gram-positives may have the equivalent of a periplasm, but they don't really have a periplasm. So.
Right. So that's a very nice story. I love it.
Yeah.
I think it's cool. I think it's straightforward.
Simple stories sometimes pay off, don't they?
I think the interesting things are that, as you said, this related mechanisms of becoming resistant to these peptides. The idea, the whole discussion of the structure of the two membranes, we really haven't talked about that much on TWiM. So that gets you into that, and we'll put some links for you for that as well. And I just think it's very interesting how you can modify these membranous structures to make yourself resistant to these peptides.
Well, bacteria are nothing, nothing if not clever, are they?
Amazing. They're really amazing. But life is like that. Life is very clever. Michael, I had forgotten to ask you a question about the first paper before we sign off here. So in that review article, they were talking about a vaccine for these group A streps.
Using the M protein.
Using the M protein. Now, you wouldn't make a vaccine for necrotizing fasciitis because it's not high enough incidence, but there must be many other diseases that would be prevented by this vaccine.
Well, the
correct?
problem there is that there's so many variants of M protein, and I'm not a vaccinologist to know how I could target a vaccine. And the other problem is that the M protein looks very similar to one of our cardiac proteins.
Yeah, you wouldn't inject M protein.
No, and so you'd have to make a very specific protein because you don't want, and the fact that there's so many different variants of M protein, even though M1 seems to be the most frequent pathogen isolated, the problem is people get recurrent strep throats, and M1, the M protein, is one of the principal virulence factors. So the whole conundrum of vaccinology and how you pick your target, which will deliver the correlates of immunity, is very dicey, especially when you think about the issues of rheumatic disease. And I just don't know enough about the vaccinology of M protein to know whether or not I would be comfortable, going down that path.
According to this, there are over 150 different M protein serotypes.
Yeah.
So they say that they're current, and there was a vaccine against 26 of these, which was safe and immunogenic in a phase 1. But now they say research is going into other antigens that are non-M antigens that eventually they think that this is something that's needed.
But this brings up a whole issue of how you begin to do those sorts of trials. And, the way we test antibiotics today is not with a double-blind placebo-based trial because that wouldn't be ethical. We do an inferiority trial where you ask the question, is the new drug inferior to the old standard of care? And so vaccines are really pretty tricky, especially when you consider the N and the fact that we just don't quite understand. One of the things I show the medical students and the dental students is the similarities in the M protein region that cross-reacts with our cardiac protein. And I ask them, is it similar? And to a person, they all say yes, because you put up the amino acid boxes and you begin to look at it. But, a straight read of the amino acids is not really what you're seeing in a three-dimensional structure that the immune system will see when you're making an antibody. So I think vaccines is still much more art than science.
Okay, well, on that upbeat note, we will conclude TWiM 35. And you have been listening to Elio Schaechter, and you can find him at his blog, Small Things Considered. Thank you for joining us, Elio.
My pleasure, of course.
Well, I do enjoy listening to you explain these things, these wonderfully complicated things. It's very, very good. And I'll see you in San Francisco in a couple of weeks. Looking forward to that.
Indeed.
Michael Schmidt is at the Medical University of South Carolina, although at the moment he is in Tennessee. Thank you, Michael.
Thank you, Vincent and Elio. Another great show.
Michael, you will be at GM, right, in San Francisco?
I will be at GM.
GM is what? Hey, you guys speak English.
General meeting of the American Society for Microbiology. It's the acronym police.
That's better.
Yeah, you should just call us out. It's fine. You're right. We use too many acronyms. I am Vincent Racaniello, and you can find me at my blog, which is at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM Communications Director Barbara Hyde and Chris Kondayan and Ray Ortega for technical help. And if you want to see what this communications department does at the general meeting, every morning they have wonderful sessions. And this year, Stan Malloy is going to be speaking with a whole range of microbiologists. And these sessions can be viewed live, or they are also live streamed on the internet. So if you go to the ASM website, you can find a link to that. We'll put a link in the show notes. And then they're also released afterwards as recordings. So really wonderful things that this communications department does. So check that out.
And since morning is actually afternoon on the East Coast, you can probably watch them at lunch live.
You bet. Good lunchtime
really good stuff.
watching.
I wouldn't miss Stanley Malloy for anything. He's such a fabulous speaker. Amazing.
Yeah, he's very good. He's going to talk with a range of people who work on bacteria and viruses.
He's going to
fungi.
turn them on like they've never been turned on.
You got it. It'll be a lot of fun. Can't wait. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.